Harvesting exosome-enriched conditioned medium from cells cultured in bioreactor flasks and isolation by sucrose cushion ultracentrifugation result in exosome preparation of high yield with minimal contaminating proteins or non-exosomal vesicles.
Using a single sucrose cushion prepared in deuterium oxide circumvents the laborious preparation of a discontinuous sucrose gradient and reduces the amount of sucrose needed to achieve the required density.
Effective in vitro delivery of siRNA encapsulated in exosomes prepared in this protocol highlights the potential of this system as a new generation RNA interference-based therapy for pancreatic cancer.
To begin, culture HEC293 cells in normal medium, as described in the manuscript. Expand the cells into 4 T75 flasks and grow until they reach 90% confluency. To prepare a bioreactor flask, add 50 to 100 milliliters of normal medium into the medium reservoir of the bioreactor flask to wet the membrane. Collect all HEC293 cells from the 4 T75 flasks and resuspend them in 15 milliliters of exosome-depleted medium in a centrifuge tube. Then use a 20 milliliter syringe connected to a blunt fill needle to carefully add this cell suspension to the cell compartment of the bioreactor flask. Then fill the medium reservoir of the bioreactor flask with a normal medium up to 500 milliliters and incubate it at 37 degrees Celsius, 5% CO2, for a week. After 1 week of incubation, remove all the medium from the medium reservoir of the bioreactor.